Okay, good. All right. Yeah. I suppose you can hear me now. So we are going indeed to be taking a look at the chapter cells as the basic unit of life. And yeah, bon, indeed, there is a part which comes before the actual study of cells, according to your specifications. We've got something called the microscope, where essentially you'll be dealing with um, microscope calibration, and essentially everything related to microscopes, and also definition between the difference between resolution and magnification, calculating of actual sizes, and the um, between comparison between different microscopes like light microscope and electron microscope. Okay, but I'm gonna be skipping those parts and just, yeah, essentially just run this very quickly. Anyway, so well, cells can be divided into two broad types, two broad categories, that is eukaryotic cells and prokaryotic cells. Now, eukaryotic cells, they have a more complex ultrastructure as compared to prokaryotic cells. Prokaryotic means like they don't have a true nucleus. However, eukaryotic cells, they are cells that do indeed have a true nucleus. Well, that's the broad categorization of cells. Now, yeah. And cytoplasm of eukaryotic cells is divided into the membrane-bound compartments called organelles. Organelles are like small organs which are found within the cells. Now, cell organelles, we are going to be rushing through all these quickly. So we've got firstly cell surface membrane. All cells are surrounded by a cell surface membrane, which essentially just separates, it just creates that barrier between the external and the internal environment. Okay, so it essentially separates the inside of the cell, the protoplasm, from the surrounding, from the external environment. Okay, and also um, cell surface membrane being partially permeable, they control the exchange of materials between the internal and external environment. So it allows the passage. How does it control? Because it is partially permeable, and hence it only allows the passage of some substances and not the other. Okay, and cell membrane is essentially a phospholipid bilayer. A phospholipid is essentially a molecule. It is a type of lipid, and yeah, I've got a lot of those phospholipid that just line up to form a layer, a sort of queue, and then you've got two layers like this, which is why we call it a phospholipid bilayer, and it spans a diameter of about 10 nanometers. Okay. Anyway, moving on. Nextly, we've got nucleus. So nucleus is present in all eukaryotic cells, and nucleus is what differentiates cells from being eukaryotic and from being prokaryotic. Okay, because nucleus is present only in eukaryotic cells and not in prokaryotic ones. Okay, so what is the nucleus? It's essentially just a large organelle that is separated from the cytoplasm. Indeed, it does have a double membrane, and it contains the DNA, which is arranged into chromosomes. Okay, and chromosomes, they contain the DNA and the proteins. Well, DNA and proteins, yeah, which are collectively referred to as chromatin. Now, chromosome, they are like just the spool and threads of DNA wrapped around histone proteins, and that's what we refer to as the chromatin. And during cell division, chromatin, by the way, it condenses to form um, chromosomes. Okay, DNA normally exists as chromatin, that is DNA wrapped around protein, histone proteins. But when the cell is undergoing cell division, it condenses to form chromosomes. The nuclear membrane is known as the nuclear envelope and contains many pores. Yeah. Now, we said that it is double membrane, right? Nucleus is surrounded by two membranes. Well, we also call, so the name for that membrane is this nuclear membrane. And we also call it, refer to it as the nuclear envelope. And on that membrane, there are many pores, which we refer to as nuclear pores. And what the importance of these pores? They are essentially just important, very important channels for the movement of mRNA, that is the messenger RNA, and ribosomes to travel out of the nucleus, as well as allowing enzymes and signaling molecules to move in. And the nucleus contains a region known as the nucleolus, which just nucleolus is just a region where the chromatin is just a bit more dense there. Yeah, here you go. Here you can see it here. And it says it is the site of ribosome production. Yeah, probably it is. Okay. Now, few important things to pay attention to. So this round figure, this red round figure, is the nucleus. Inside the nucleus, the darker region here is called the nucleolus. Inside the nucleus, you've got chromatin. The membrane surrounding the membrane that forms the outer the membranes of the nucleus is called the nuclear envelope or the nuclear membrane. It is a double membrane comprising of an inner and an outer one. Chromatin, yeah. Now, why was I emphasizing on an outer and inner membrane? Because actually the outer membrane of the nuclear of the nucleus, the outer membrane of the nucleus actually extends to form the endoplasmic reticulum. Well, more precisely, it is the rough endoplasmic reticulum. We'll take a look at that shortly. So rough and smooth endoplasmic reticulum. So the endoplasmic reticulum, which we often write in the shorthand form ER. So it is made up of a series of membranes that form flattened sacs within the cell cytoplasm, and these sacs we call the cyst. We refer to them as cisternae. So the ER is linked with the nuclear envelope. Well, it's more precisely the outer membrane, the outer layer, because it is double membrane. It's the outer layer that is linked, that is continuous with the nuclear with the ER, the RER. And there are two distinct types of ER with different roles within the cells. Now, their roles, their functions is crucial. Understanding of their roles is crucial. So the rough endoplasmic reticulum, the RER, it continues folds of the membrane that are linked with the nuclear envelope. So the surface of the ER is covered in ribosomes. So as you can see here, on this endoplasmic reticulum, there are just little dots, and these little yellow dots are ribosomes. Now, why the fact that the ribosomes on it, under the microscope, it sort of look rough and packed, not sort of patchy, but essentially rough. And it is from here that the word is derived, rough endoplasmic reticulum. So, and this is why we call it the rough endoplasmic reticulum. So the role of RER is to process proteins that are produced on the ribosome. So why do you think there's a close link between ribosomes and the endoplasmic reticulum? Is because they must be having a sort of interconnected, they must be working sort of interconnectedly. So yeah, so actually the proteins that the ribosomes synthesize is further processed by the RER. Okay, now yeah, they may add stuff, anyway. So the smooth endoplasmic reticulum, now the SER, it does not have any ribosome on its surface, and which is why we call it smooth, because under the microscope it just looks smooth like this, the yellow orange ones here. Okay, and it is, and the SER is involved in the production of lipids and steroid hormones like estrogen and testosterone. And here you go. Here's a big picture of all the things. So here got nucleus, outer membrane is continuous with the rough endoplasmic reticulum, and then it is rough because there are ribosomes on its surface. And then as we extend, as we go farther away from the nucleus, we the area is rather the endoplasmic reticulum is rather is smooth here because there are there are no ribosomes on their surfaces. Okay, and they are more tubular like, whereas those ones are more sac like. Those cystini are more sac like, and these cystini are more tubular like. Okay, and now moving on to the next structure is the Golgi body. Now the Golgi body is often referred to as the Golgi apparatus or the Golgi complex. It consists of a series of flattened sacs similar to those here. These are also flattened sacs. Here also it's a flattened sacs, and of course it's membrane bound, and it can clearly be distinguished from the ER because they are not interconnected. Okay, they are not interconnected, whereas the endoplasmic reticulum is actually connected in close proximity to the nucleus. Now, it can also, it is not connected to the membrane bound compartment. So as you see here, the cystini are connected with each other in it, but here they are disconnected. Actually, there's no connection. They are just in close proximity, but they are just suspended independently. They are not connected. Okay, and they have a distinctive Wi-Fi symbol sort of appearance. You know that Wi-Fi symbol sort of, yeah. And here we call it a cis phase, and here the trans phase. Its role is to modify proteins and package them into vesicles. Now, package them into vesicles is a very important word, and it is a word that you should use when you are asked to describe the function and the role of the Golgi apparatus. Its role is to modify proteins and package them into vesicles. Okay, so you see those vesicles forming, okay, pinching off from the structure. So yeah, they are just packaging proteins after having them modified, the packaging, so for transport. Okay, and yeah, these are called cystini. Here it receives vesicles. It receives vesicles that buds off from the endoplasmic reticulum. Yeah, there are some that bud off, and here it buds off its own structure to transport proteins. Next, you've got the mitochondria. The mitochondria, which in singular form we call it mitochondrion. If we are referring to only one, then we say mitochondrion. And these structures are relatively large organelles and surrounded by double membrane. Again, they are smaller than the nucleus and chloroplasts, but they can be seen with the light microscope. And so here you see there are two membranes, an inner membrane and an outer membrane. And the inner membrane actually fold in. Okay, you see these fold, they're just sort of invaginations that we refer to as cristae. So cristae are those fold convolutions, okay, essentially. And what the function of mitochondria was it here? It's actually a site of aerobic respiration, or as we say, the oxidative phosphorylation, oxidative phosphorylation, oxidative phosphorylation, and within eukaryotic cells. Exactly, they are present in eukaryotic cells. And the mitochondrial matrix, so the liquid present inside with that contained within the inner membrane is called the matrix, and it contains enzymes that essentially catalyzes the reaction, the aerobic respiration. Because aerobic respiration, don't forget, is a enzyme controlled reaction, enzyme catalyzed reaction. Now moving on, small, yeah. So the mitochondria actually it has its own DNA as well. So it's called small circular pieces of DNA, which we refer to as the mitochondrial DNA. And ribosomes are also found in the matrix. By the way, we've got two types of ribosomes: 70S and 80S. So mitochondria they contain the 70S ribosome, the smaller type, and this allows for the production of protein required for respiration. So the fact that it's got its own DNA and own ribosomes mean that it can make its own proteins that it will be requiring for respiration. Okay, so yeah. Ribosomes, they are found in the cytoplasms of all the cells or as part of the rough endoplasmic reticulum in the eukaryotic cell. So yeah, we've got two types of ribosomes, which we call free ribosomes or free ribosomes. They are just found free lying in the cytoplasm. Otherwise, you've got ribosomes that are attached to the endoplasmic reticulum to form part of the rough endoplasmic reticulum in eukaryotic cells. And each ribosome is a complex of ribosomal RNA and proteins. Okay. And we've got 80S ribosomes, which are composed of 60S and 40S subunits, and they are found in eukaryotic cells, as compared to 70S ribosomes, which are found in prokaryotes like mitochondria and chloroplasts. Well, actually, mitochondria and chloroplasts are considered to be prokaryotes, but yeah, it's sort of a theory. So yeah. Anyway, ribosome. The function of ribosome is just the site of translation during the process of the protein synthesis. Now, why translation? Because it essentially translates. Okay, you've got mRNA, the messenger RNA, and it essentially translates this message to make proteins uh, with the help of tRNA. Okay, and now you've got vesicles. Vesicles, they are essentially small membrane-bound sacs used by cells for transport and storage, and they can be pinched off by the end of the Golgi body, and these are known as the Golgi vesicles. And they can fuse with the cell membrane to allow exocytosis or form the membrane during endocytosis. So exo means the exit of substances, so the outward bulk transport of materials through the cell surface membrane, and endo means inside, in. So it's the engulfing of substances into the cell, the fluidity, blah blah blah. Okay. Yeah. Lysosomes. Now, lysosomes, they are specialized vesicles. They are similarly, they are vesicles as well, but they are more specialized vesicles because they contain hydrolytic enzymes. Now, the fact that they contain hydrolytic enzymes mean that of course, they cannot be made from just the normal cell membrane, phospholipid bilayer, phospholipid layer. Okay, they are made from different materials. And hydrolytic enzyme, they break down, well, they essentially hydrolyzes substances, break down biological molecules, example, waste materials such as worn out organelles, engulf pathogens during phagocytosis, and cell debris during apoptosis. Okay. It contains 60 plus types of enzymes here contained in this small structure. Now, we'll soon be wrapping up, but here we've got centrioles. Centrioles, they are just hollow fibers made of microtubules. And we've got two centrioles at right angle, or we said they are orthogonally arranged. That is, they are arranged at right angles to each other to form a centrosome, which comprises the spindle fibers during cell division. Centrioles are not found in flowering plants and fungi. And now microtubule, that's something interesting you need to be knowing. So microtubule, they are hollow tubes. So micro means small and tubules essentially small tubes. So microtubules are small tubes or small hollow tubes made of tubulin protein. Okay, they are made of the protein tubulin. So we've got two types of tubulin proteins. We've got alpha tubulin and beta tubulin. Well, actually, we've got gamma tubulin as well, but we're not really exploring it in this in our syllabus. Anyway, so alpha tubulin and beta tubulin proteins which combine to form a dimer. Like you've got polymer, and here you've got two monomers. Okay, so we call it dimer. So, and then they are further joined together to form a protofilament. So you see, let's suppose this here's an alpha tubulin, here's a beta tubulin. They combine, they interact to form a dimer. Now we've got several dimers. They've got several dimers that just attach to each other to form a protofilament. And then, so protofilament is like one of this long chain. And then you've got 13 protofilaments that interact to form a cylinder. So this cylinder now got 13. So if you, this is top view sort of, so you will see there's 13 protofilaments. Okay, and microtubule, they make up the cytoskeleton of the cell, along with I forgot those names. You've got a sort of fiber which are okay. Anyway, either way. So microtubule make up blah blah blah, support and movement. So yeah, obviously it supports the shape the shape of the cell, and but it also allows a protein. So we've got that walking protein, you know, so transfer RNA, transfer protein. I think it's myosin. So it just works on this to transport substances. Yeah, by the way, we've got also intermediate filaments, and you've got actin filaments, which also forms part of the cytoskeleton. But intermediate filaments and microfilaments, that is actin and intermediate filaments, are not really within the scope of our syllabus. Okay. Now, lastly, we've got cilia, hair-like projections, very boring. Microvilli, increased surface area. And yeah, do not confuse microvilli with villi. Villi are larger structures, by the way. This microvilli is micro, it is at the cellular level. It's extension of the cell membrane. Okay. Cell wall, that's very boring. It's made up of beta glucose, which are 180 degrees rotated. You've got chloroplast. Well, we'll be exploring chloroplast a bit in more details in the chapter where we deal with photosynthesis and stuffs. We've got plasmodesmata, also something that you'll really be dealing with in another chapter rather. We've got a large permanent vacuole in plant cells. Okay, so we are done with the cell structures, cellular structures essentially, and I'm going to pause a bit here because yeah, it'd be too overwhelming if I just put all data at once. Now let us digest data a bit by a bit, okay, small by little, and yeah. So right now what we've seen essentially is eukaryotic cells. Now you know that animal and plants are both eukaryotic cells in it. Yeah. So okay, just a quick picture here. So how do electron micrographs look? So exam question will not always contain neat diagrams or cellular structures, but may instead present images taken using microscopes, which are called micrographs. Now you need to be able to identify organelles in micrographs based on the shape, location, and size relative to other organelles. Okay, so something like that. You need to know the structures. Well, they don't look as hideous as this one they've put here. They look the real one looks much more better. Anyway, all these pictures are very boring, so I'm gonna move on now to perhaps some few exam questions. Now I'm gonna leave you to take a 1 to 2 minute of break. In the meanwhile, you try to digest information while I will present shortly the few exam questions. All right. So yeah, I'm gonna leave you. Take a break for 1 to 2 minutes. Then we'll come back. Okay. So we are back, aren't we? All right. Now let us take a look at this question. So it's about apparently it's a question of May-June 2021, and it's the paper 2, the first variant of paper 2. Now, the question is rather interesting because we don't really go in that much details in the notes, but as you worked out the questions, you will learn through the mark scheme. So I've included the mark scheme answers there, so we will take a look. Now, lysosomes or cell structures. Well, we did go through the structure briefly on lysosomes because in the notes they are not as detailed. But essentially, they are cell structures that contain enzymes that are known as high acid hydrolases. Okay, we said they contain hydrolytic enzymes, essentially the same. Okay, yeah, and figure 6.1, that is this picture, this whole picture here, it shows some processes that occur in animal cells. Okay, now we see here it says bacterium, so there's bacterium on the, well, supposedly this is a cell, so there's bacteria outside the cell, and it is just taking in the bacteria. It's taking in the bacteria and processing it, just enclosing it within a membrane, and then seems like it's digesting it in it. So this process is what we often refer to as phagocytosis in it. So yeah, this engulfing bacteria. So yeah, phagocytosis. Now let us try to identify a few structures. So here it says lysosomes, that is those vesicles, special vesicles that contain those hydrolytic enzymes. Okay, and here, what's this structure, by the way? Yeah, separated cysteine, separated cysteine. This structure is called our, yeah, it is our one and only Golgi apparatus. So you may say Golgi apparatus, Golgi body, or Golgi complex, or whatever. And here you can see vesicles just budding off from the Golgi apparatus. They are budding off and they are fusing with this phagocytic vesicle. They're fusing, and then the enzyme will just sort of digest the bacteria in it. So yeah, and we can see some vesicles that's budding. Well, what's this structure, by the way? You've got a little dot on it. It's rough indeed. It is our one and only, the trusty old rough endoplasmic reticulum in it. And we can see some structures that are budding off from the rough endoplasmic reticulum and coming here to fuse with the Golgi apparatus. Okay, and what around here? We see like it's just mitochondria, isn't it? It does not look like a mitochondria. I've got a double membrane and an inner convoluted membrane forming the cristae. But it does not seem too normal. It seems like defective, doesn't it? It should normally just be like this. Let me draw a proper one. It should be somewhere like this. Okay, and then like this. Okay, but here it just seems a bit defective out here, right? So perhaps they are just killing it because it's defective. And this you'd say autophagy. Okay, so this is what we call as autophagy. Now autophagy, this is a special type of autophagy which whereby the mitochondrion is being killed. Okay, now name the cell structures labeled. Just give me a minute with you. Yeah, let us proceed now. So here there was a question. Let us see what were the questions. So name the cell structures labeled A and E. So let us go and take a look. What is A? The A is our rough endoplasmic. E is our Golgi body. So here you go. Rough endoplasmic reticulum and Golgi body or apparatus or complex, whatever you want to call it. So the function of structure label F. Let us see. What is F? Okay, these are vesicles that's budding off from the RER. So they are just transporting the processed proteins to the Golgi body. So we state the function of the structure. So these vesicles, they transport modified proteins from A to E. You may say from A to E or from rough endoplasmic reticulum to the Golgi body, from rough endoplasmic to Golgi body, however you call it. Name the process by which bacteria are taken into the cell. So you may say phagocytosis or endocytosis because it's coming into the cell membrane. So endocytosis, whatever you call it. With reference to the processes occurring at B and D in the figure 6.1, outline the role of acid hydrolases in lysosomes. With reference to processes at B and D. B and D, okay, here's killing digesting bacteria, here's digesting an organelle. So I said, with the help, with the reference to outline the role. So it helped in digestion of bacteria at D, and it helped in digestion of defective mitochondrion at B. During the process called the autophagy, in the process, the myriad of enzyme that comprises the lysosomal acid hydrolases catalyzes the substrate like proteins, phospholipids, and nucleic acids. And the resulting biological molecules are then reused, recycled within the cell. And that is your full answer for 3 marks. Okay, here you go. And this is your mark scheme, by the way. And yeah, obviously, I took the answers from the mark scheme. Yeah, break down, digest bacteria, digest defective organelles, or you may say mitochondria, you may say autophagy, is catalyze or hydrolyze whatever biological molecule should be named, reuse or recycle of biological molecules. Okay, cool. Now next, yeah, by the way, when I talked about the mitochondria. Now, why did I say where those all biological molecules come from? Well, because the organelle mitochondria in itself is composed of mitochondrial DNA in it. It does have its own DNA, doesn't it? It is a double membrane organelle in it, so it does have a phospholipid bilayer, and we do know that the hydrolase does contain phospholipase, so this is digested, so we get phospholipid. In its cell surface membrane, there are proteins that are embedded, so those proteins are digested to get amino acids. And concerning its mitochondrial DNA, it contains deoxyribonucleic acid, and our hydrolytic enzyme, it contains nucleases, so this is also digested. I think it is. I suppose it is the same question, different parts. Carrier proteins in the membranes of lysosomes. In the membranes, yeah, okay. So yeah, it's talking about these sort of proteins that are embedded in the membrane of lysosomes. They maintain a lower pH than the surrounding cytoplasm by moving hydrogen ions. Now, suggest how the carrier proteins maintain the lower pH within the lysosome. Because remember, it needs an acidic environment to contain all those hydrolytic enzymes in it. So actually, they are proton pumps that move hydrogen ions into the lysosome against a concentration gradient, that is by active transport. That is using ATP from a respiration. Well, you get your 2 marks using only say move hydrogen ion inside and refer to active transport, but I extrapolated a bit more, and that's fine. I need to give you the best answers in it. Ok, maintenant Golgi body. Let us take a look at another question. The Golgi body, rough endoplasmic reticulum and smooth blah font partie de l'internal. Maintenant, membrane, il y a une fluide mosaic structure, pas besoin qu'on soit là pour le moment, et des autres matériaux, oui. Ok, je vais montrer une photo, allez. Là, tu as mitochondria, l'infinite diète, mitochondria, ça. Tu vas trouver inner convoluted structure, tu vas trouver ce double membrane, inner convoluted structure. Ça, là, les vertically, tu mets comme ça, tu coupes là, tu vas guetter depuis là-haut. Mais par contre, ça là, les tient endormi comme ça, après tu coupes tu vas guetter depuis là-haut. Mais c'est tout le mitochondria, même ça c'est une mitochondria, ça aussi une mitochondria. Tu vas trouver un point, Laura. Ça c'est comment structure ça? C'est au AIA, rough endoplasmic. Sa structure là, alors qui peut être, l'écoute smooth endoplasmic, parce que les continuous with this. Oui. Alright. Next, is to describe the difference in the structure and function of SCR and RER. Alors, tu sais que RER, est tout simplement process protein qui a besoin de former l'oli, et SCR, les forme lipids lipid synthesis, okay? It produce lipid synthesis of lipids and steroids like estrogen and testosterone. So difference in the describe difference in structure and function. Alors, structure. Auriole, il y a un ribosome on the surface. SEOL, bena. Auriole, is continuous without a membrane of nuclear envelope. Mais par contre, SEO, non, il n'est pas directement continuous. Et auriole, il y a un arrangement un peu plus régulier. Mais par contre, l'autre là, il Ce fonction, auriole, il produce ou bien de cas transport protein. Mais process aussi, il ajoute une structure à une protéine là, comme glycoprotein. Et ainsi, il produit lipides. Il délève avec lipides, cholestérol, stéroïdes, tout ça. Ok, that's it. 3 points bien faciles. Voilà, ça c'est chose là. Mock scheme là. Answer, which you may, you know, if it helps. More question. Ok, ça question là, il parle de phospholipide. Bon, on ne dépend qu'au faire phospholipide. Mais il bon, the target and cells and growth overview. Il a describe structure d'un phospholipide molecule. C'est quoi un phospholipide? Il a un phospholipide, un triglycéride. De faire triglycéride et un glycérol avec 3 fatty acides. Non, n'est-ce pas? Alors pareil, les différences entre phospholipides, c'est quoi? Les déplacer dans 3 triglycérides nazis, pas triglycérides, déplacer dans une étiquette qui est dans un triglycéride et dans un glycérol avec 3 fatty acides. Mais phospholipides, il y a ce glycérol pareil, mais il y a des fatty acides. Et sa troisième là, il est remplacé par un phosphate groupe. Ok? Alors, parce que phospho d'ailleurs, phosphate. Alors, phospholipid molecule is a type of lipid comprising a hydrophilic head containing phosphate group and 2 hydrophobic fatty acid tail that are attached to a glycerol molecule. It may contain an additional variable R group, example choline. A fatty acid tail may be saturated or unsaturated. Ça me finit comme summarize antiphospholipide en segment créole. L'autre question maintenant, très intéressant, toujours bien important, qui travaille sur cette question-là parce que c'est ça qui complémente your notes. The notes, c'est pas, it doesn't mean everything, et bien sûr, il dépend. Par exemple, si moi, je vous donne un note, bon, pour finir, inclure tout ça va affaire important là-dedans. Mais par contre, moi, pas quoi, tu dois vous préparer. Car je vous donne toi, note c'est directement dans le livre, mais dans le livre, là, il peut y avoir une réponse mark scheme, tout ça là. Alors, il est important que tu travailles avant d'affaire. Anyway, the one function of Golgi body is to package. Alors, ça, mon package, c'est important. Is to package molecules into Golgi vesicles. A Golgi body and a Golgi vesicles are not visible in figure N.N. Est-ce que vous incluez une figure N.N ici? Ok, c'est une photo là. Hein? Ok, apparemment. Ok. Et de toi, describe the features other than the presence of Golgi vesicles that will help you identify a Golgi body in a transmission electron micrograph or another area of the same liver cell. Other than the presence of Golgi vesicles. Ça veut dire comment tu connais qui features l'autre que ce vesicle qui pourrait pas identifier Golgi body. Mais tu vous trouves structure là, tu vous trouve ce van. Essentiellement, les visiteurs describe and Golgi body. Les visiteurs essentiellement describe and Golgi body ce structure. Mais c'est quoi? It consists of flattened sac called cisternae with a smooth, de vous identifier d'après ce smooth outer surface parce que les ribosome, s'il y a un ribosome, veut dire s'il y a un ribosome, vous êtes, veut dire rough endoplasmic reticulum. Mais il y a un cisternae et les ribosome. Mais là, les cas SIA, parce qu'il y a aussi ribosome et il y a un cisternae. Mais non. Ok? Ici, chaque cisternae, il y a un stack qui est séparé. Il y a un stack qui est séparé. SIA, les collés, mais Golgi body, ce van système, il est séparé. Veut dire, il n'est pas interconnecté. Ils sont interconnectés et toujours les prénucléus. Et en addition, chaque système, il y a une solen, il y a une structure un peu comme ça. Alors, moi, tiens, comme un red blood cell. Red blood cell, là, c'est une exagération, mais il y a un peu comme ça. Okay. Yeah, regarde là. Tu peux trouver ça, c'est une solen ends. Dans ce bout-là, il y a un peu plus gros. Alors, c'est ça, c'est ce qui te besoin expliquer en ce qui te lorsque tu expliques le Golgi body. Alors, flatten stack in Marvel System, il stack ou bien layered, et il séparé, il n'est pas interconnecté, il smooth, no ribosome, close to nucleus, single membrane, soul and ends, two distinct phases, donc il y a 6 phases et the trans phase. Et yeah. Okay, some Golgi vesicles may contain secretory proteins. Vesicle pour contenir protéines. Ok, oui. C'est ça la fonction de travailler une protéine depuis Golgi. Ce qui finit process. D'ailleurs, pour ça, il y a amené de côté code design. Okay. For release from the cell. Describe the sequence of events that occurs following the packaging of secretory protein into a Golgi vesicle that its release from the cell. Alors, Golgi body. The Golgi vesicle, they move along the surface of the microtubule to the cell membrane. Et c'est tout simplement ça qui arrive. Quand il finit package, qui va arriver? Le vesicle là pour bouger pour aller côté cell membrane. Là, qui va arriver? Il pour fuse avec cell membrane, les protéines of release, 3 points. Qui veut dire any free from. Alors, qui m'en cause? Cause vesicle move to the membrane. The movement of vesicle, c'est via the microtubule. Et dernièrement, les fuse avec cell surface membrane to release the protein. C'est ça. Correct? Et dernièrement, une question intéressante. Some Golgi vesicles contain glycoprotein and glycolipid can be added to the cell surface membrane. After the role of glycolipid, c'est act as receptors. Ok, ça c'est le chapitre, mais moi je finis par lien. Et déjà matériel. Ok, moi j'ai une question. Est-ce qu'il y a des autres questions que je travaille pas ici? Ok, non, ça c'est enzyme. Ok, ça va nous être c'est enzyme. Mais I'll work on those. I'll work on more questions for you, and perhaps in another session we'll take a look at those. Ok? Allez, au revoir. Au revoir et on se voit très prochainement pour une autre vidéo.